Thank you for the opportunity to come and tell you a little bit about what we're doing in personalized medicine. As you heard, I started my career as a clinical pharmacologist, and I increasingly define myself that way still. And I've been interested in antiarrhythmics, so I won't say much about arrhythmias at all in this talk. But about 4 years ago, I gave a talk in clinical pharmacology, and one of our senior faculty said, that pharmacogenetic stuff is pretty interesting. Why aren't we actually doing it? And so that sort of serves as an impetus to the kinds of thinking that I'm going to expose you to. These are two statues on the Vanderbilt campus that I walk by every day as I walk from the parking garage to my office. So it sort of gives you a sense of, maybe this sort of inspires me every day. I collect titles, and we were talking about that at dinner last night. I'm glad I didn't include all of them on this slide. So this doesn't, the bottom one. Okay. I was trying to go backwards. So personalized medicine is what I am trying to think about all my waking hours right now. And personalized medicine is more than just getting genetics to the bedside, although that's what I'm going to focus on. So I want to say at the beginning that personalized medicine involves more than just genomic medicine, but it involves personalizing a health record, getting patients to engage in their own health care, getting the community to engage with us in care, improving literacy and numeracy, individualizing therapies, all the things that are on this slide. So I say that not to dismiss them, but to say that I just don't have time to talk about all those kinds of aspects of a program in personalized medicine. So what I'm going to talk about is listed here. You can read it. It's a little bit of a couple of stories just to sort of set the stage, a little bit about where we are with personal genomes right now, and then I'll talk about BioView and how we set it up and what we're doing with it, and how we hope it's going to be a resource, at least for us and perhaps for a wider community. So if you talk about genetic disease, I don't know why I chose this particular slide. This is not a disease I work on a lot. This is hypertrophic cardiomyopathy. This is, and the notion is that in 1990, you could define a genetic disease by a phenotype, or we were starting to get to the point where you could define disease genes. So in hypertrophic cardiomyopathy, there are now 15 disease genes, and in many of the cardiac ion channelopathies that I study, like congenital long QT syndrome, there might be, there are again sort of 12 different disease genes, and it's a great story. In translational medicine, of identifying families, identifying mutations, understanding the relationship between mutations and families, understanding how that affects prognosis and therapy, and I'm not going to talk about that at all. So, but that's genetic diseases, and you'll hear a lot about that from Dr. Gall later today, right? Right.
I'll try.
Okay. So this is one of my favorite quotes. I don't know how I stumbled across it. Paul Dudley White is one of the founders of American cardiovascular medicine, and he wrote in 1960 in Circulation that atherosclerosis runs in families. The mechanism by which this inheritance occurs is still a mystery. So it still is a mystery, but the mystery is being unraveled. The mystery is being unraveled by the technique of genome-wide association, at least now, and more generally, by an in-depth look at what the genome contains in families that have premature atherosclerosis. So I'm not going to walk you through the technology of genome-wide association, but it is possible to take, in this particular slide, about 2,000 individuals with premature coronary disease and 3,000 individuals without premature coronary disease, interrogate each one of them at 500,000 to 1 million common SNP sites, common polymorphic sites across the genome, and then ask the question, are there any sites in that interrogation that associate with the phenotype of premature coronary disease? So it's genetics at a different level. And the way these data are displayed, as you know, are the p-value for the individual chi-square comparisons at each one of the 500,000 SNP sites is on the y-axis, the chromosomal location of the individual polymorphism is on the x-axis. And I'm fond of saying here, it does not take a geneticist to tell you that there's something going on here. This is probably the most replicated signal in genome medicine. This is a technology that is a grand total of 4 years old and has been used to approach many, many, many diseases. This is probably one of the furthest along examples. And so the chromosomal signal at 9p21 replicates over and over and over again when you look at patients with premature coronary disease. What's really interesting about this signal is that it's nowhere near any known gene, and no one has any clue fair to say, what the underlying biology is. So I'm fond of saying there's a biomarker in here, there's a drug in here, there's intellectual property in here for sure, but beyond that we don't know what there is in here. So that's an example of new advances. And I guess I should also say that if you carry one of these alleles, these are common variants, so if you carry one of these alleles, 30% of you do, your risk of coronary disease before the age of 60 is increased somewhere between 25 and 50%. So it's not like carrying a mutation that gives you hypertrophic cardiomyopathy where your risk is increased 1,000-fold. This increases it 1.5-fold. So that's important because if I told you you had a variant like that, you shouldn't get too excited and you shouldn't go out and double your life insurance or commit suicide or something like that. So we are in an era where we're moving from single variant genetics to multivariant genomics. There are many definitions of the differences between these terms, but this is one of them, and we concocted this particular graph for a presentation from the Pharmacogenomics Research Network, of which I'm a part. And actually, I should say that, of course, when we published this, it was the Pharmacogenetics Research Network, and now it's the Pharmacogenomics Research Network, because the work that we do within the network is designed to try to understand the nature of variants that affect variable drug responses. And you can think of variants as one at a time, one variant conferring a very large effect, or you can think of many variants that in total confer variability in drug response. So that's the evolution from pharmacogenetics to pharmacogenomics that our field is seeing. So when I talk about pharmacogenetics and variability in drug responses, people often think of these kinds of phenotypes. And if I knew any of the students in the audience, I'd say, tell me what these are. So what they are this is a case of ACE inhibitor induced angioedema. This is rhabdomyolysis due to a statin drug. This is an intracerebral hemorrhage due to whatever anticoagulant you want to choose. Stevens-Johnson syndrome due to some drug exposure, Tegretol or something like that. Hemolytic anemia in a patient with G6PD deficiency exposed to an antimalarial drug, usually African American. And this is the most important one of all, drug-induced torsion decline. That's what I study. So I think that's the only arrhythmia thing that I have to say. So that's what people think about, and there is lots and lots of interest in trying to define the genetic basis of these kinds of adverse drug effects, because of course if you could predict who was going to develop these catastrophic kind of things, you might be smarter in terms of avoiding certain therapies in patients or in terms of constructing drugs that didn't do this. But the real problem, I think, in modern therapeutics is illustrated by this slide here. These are 4 examples chosen from work that goes on in the Pharmacogenetics Research Network. And the 4 phenotypes are not terribly interesting. I mean, you can read what they are, but the point is that if you look at large populations of patients exposed to common diseases, common drugs, what you see is tremendous variability in the response to those drug therapies. And I'm fond of saying that the commonest adverse effect of contemporary drug therapy is that the drug doesn't work. And you can look at surveys of cardiovascular drugs, of antidepressant drugs, of anti-cancer drugs, and efficacy rates are never 100%. They're always much less. They're always less, and sometimes much less than that. So the notion is, if you can understand the nature of variability in response, you may be smarter in terms of, again, prescribing current drugs or developing new drugs. I think some of this has to do with pharmacogenetics. The same dose doesn't produce the same drug concentrations in each individual patient. And some of it has to do with variability in the diseases that we treat. So when you treat, nobody in their right mind would take seriously a doctor who said, well, you have jaundice, and the treatment for jaundice is X. That's the way people practiced medicine 100 years ago. So nobody in their right mind now would accept the idea of somebody saying to you, you have cancer. The treatment for cancer is X. Well, we now know that there's many, many different molecular subtypes of cancer. That drives what your choice of therapy is. And by the same token, nobody should take very seriously somebody who says, well, you have atrial fibrillation, or you have hypertension, and the treatment for those diseases is X. So there's an intimate relationship between disease genetics and pharmacogenetics that remains to be largely explored. I'm going to give you a couple examples from pharmacogenetics. to set the stage for what I'm going to talk about later. So this is a picture from the very first paper that I ever published. We were fortunate enough to be working with an antiarrhythmic that most of you hopefully don't remember, called Encanide. And we studied 11 patients, and in the finest tradition of translational research. And I'm not being totally facetious about that. And we were interested in, these are patients who were selected because they had high-frequency ventricular ectopic beats. And we gave each of them increasing doses of Encanide until we established whether or not they would respond to the drug by suppressing their ectopic beats. That's the way we did it back then. But at the beginning of the study, we did something interesting because it was a real translational science exercise. We gave each patient a single dose of drug and then followed pharmacogenetics for 24 hours. So you can see that we had 11 subjects, 10 of them responded to drug. I don't want to go into what the responses were. They were electrocardiographic as well as other things. And they were characterized by very rapid drug elimination and very, very variable drug concentrations, from 1 to 100 nanograms per ml. So pretty impressive response. And then we had one non-responder. We figured her concentrations would be very low, but when we actually got the data back, her concentrations were much higher than anybody else's. The doses she got to were much higher than anybody else's, and she had very, very slow elimination. So it took us months and months and months of the greatest minds in pharmacogenetics to figure out that, in fact, Encanide is a prodrug, and it was biotransformed in these patients to a very active metabolite. That's why they have rapid elimination. This patient is one of the 7% of this audience who's homozygous null for a mutation or a variant in cytochrome P450-2D6, CYP2D6. So she failed to biotransform Encanide to its active metabolite and therefore failed to display a response. Encanide, for reasons that I could talk about for hours and hours, has now gone away. Probably a good thing. But other CYP2D6 substrates are out there. The most notable one, and this is a story that is evolving, but I think it's probably real, is Tamoxifen. Tamoxifen is probably a prodrug. Its effects are largely mediated by biotransformation, mediated largely by CYP2D6 to other metabolites. This is one of them. And if you examine outcomes of patients treated with Tamoxifen as a function of genotype, it turns out that patients who are extensive metabolizers, who make a lot of this, have better responses than patients who are poor metabolizers. The really interesting and potentially important part of this is that a group of drugs that inhibit CYP2D6, so that would be predicted to reduce Tamoxifen effect, are SSRIs, many SSRIs, notably fluoxetine and paroxetine, Paxil and Prozac. So, and it's pretty common for patients on Tamoxifen to take those drugs because a common side effect of Tamoxifen is flushing, and those drugs do a great job in reducing the flush. Probably they do a great job in reducing the flush because they do a great job in reducing the pharmacologic effect of the drug altogether. So there are now data, these are data from Mayo and Indiana, but there are now data from Medco, the pharmaceutical benefits company, that says that it really makes a huge difference if you put somebody on one of those SSRIs in terms of their recurrence rate during therapy with breast cancer. A lot of people don't use Tamoxifen anymore because aromatase inhibitors are better. But if you look at the data comparing aromatase inhibitors to Tamoxifen, there's not much difference. And if you could parse out the ones who are destined not to respond, Tamoxifen is as good as or better, and certainly a lot cheaper. So I want to say a little bit about Warfarin, the current poster child for pharmacogenetics. I think, I'm not sure how that story is going to play out. But we've been very interested in Warfarin, and there's a point to this, and there are many points to this story. So if you asked a clinical pharmacologist 10 years ago, what is it that drives variability in response to Warfarin, they would have said variable metabolism, the enzyme that does that metabolism is cytochrome P450-2C9. And there are common variants in 2C9 that are all loss of function, reduction of function or loss of function variants. So if you have one of those variants, you are more likely. So if you compare patients taking a very low dose of drug at stable dose, they're much more likely to have one variant or even two variants compared to a group of clinic controls. This is a very, very small study from England. Really hasn't been well replicated since. There's no newer data than this that speaks to this. The really interesting part of this is the major bleeding part. There's lots of data that speak to this part. But the major bleeding part is that patients taking less than 3 milligrams a day tend to have a much higher bleeding rate, even during chronic therapy. And that's an interesting observation that I can talk about later if someone's interested. But the point is that the genetic variants drive variability in response to drugs, and all was well in the Warfarin pharmacogenetics field until 2004, when a group of investigators studying a really, really rare disease. So this is, again, when Dr. Gall talks, he talks about all these diseases that you've never heard of. Pay attention, because sometimes really rare diseases are the introduction to something really, really important and common. So there's this rare syndrome called warfarin resistance. You give patients hundreds of milligrams of warfarin and nothing happens to their INR. The reason nothing happens is they have loss of function mutations in the coding region of a gene called VESPA, which is the warfarin target. So if you don't have the warfarin target, warfarin doesn't do anything. It's part of the vitamin K apparatus. So people immediately said, well, maybe variability in VESPA accounts for some of the variability in warfarin response. People looked in the coding region, not much there in the general population, although I'll come back to that in a second. But in the coding region, there are a couple of common polymorphisms in the promoter, and those drive variability in response to warfarin. There's lots of data on that. I'll show you our own, of course, that if you look at initiation of therapy, variants in VESPA make a difference in terms of response, the immediacy of the response to warfarin, whereas variants in CYP2C9, which are important in determining steady state warfarin dose, don't make much of a difference in the time it takes to get to steady state. There are lots of data, much of it much better from something called the International Warfarin Pharmacogenetics Consortium that we participate in. But I just show you this as a segue to the next slide. So this slide looks at a group of patients who are receiving very high warfarin doses in an anticoagulation clinic. They looked at 200 patients. In 180 patients, the explanation for the very high warfarin dose requirements was, no surprise, non-compliance. And that's always an issue in pharmacogenetics. But the remainder were compliant, and they were compliant because people could actually measure serum warfarin concentrations on this complicated slide. But basically what this slide shows is that if you look at those patients who have high serum warfarin and yet not much budge in their INR, you find coding region variants in VESPA. So this is the segue between common variation in the genome that I can sort of look at this audience and say some of you have CYP2C9 star 2 or star 3, and these are really rare variants, 1 in 100, 1 in 1,000. Now they're rare, except you have to ask what ethnic group they're looking at. So the one that I think is the most telling is this one, totally undetected in any population except Ashkenazis. In the Ashkenazi Jewish population, if you look at patients who receive high doses, it's a common variant. So I don't know whether the right word is mutation or polymorphism, and I'm not sure I want to get excited about the difference between the two. So in genome science, what one can do is one can construct this kind of graph, and what it does is it relates relative risk conferred by having a variant to the frequency of the variant. So what we have are really rare diseases like hypertrophic cardiomyopathy, like some of the rare variants in CYP2C9, perhaps the VESPA variant. Those are rare and important to you if you have them. So if you have a hypertrophic cardiomyopathy mutation, it's important to you, but if you're a population scientist, if you're an epidemiologist looking at death rates across hundreds of millions of people, who cares about this? On the other hand, if you have the 9p21 variant, you as a person, it doesn't much matter to. But if you're a population scientist and you suddenly have a group of people who have a 40 or 50% increased risk of myocardial infarction, that's a big deal. And the same with some of the common VESPA variants. And a lot of where we're going to end up, I think, is this idea that there are going to be common and rare variants that are somewhere along this spectrum of risk versus allele frequency. And it'll be really interesting to sort all that out. So how do you do a genetic test right now if you're going to prescribe a drug based on genetics? You order a kit from a company, and then you get your pathology lab to run that kit, and a couple of days or a couple of weeks later they'll give you a result. No one is going to pay for it, and by then the patient's already on the drug that you want them to be on. So that's the way we do it now. In 2000, there was this cartoon in the New Yorker, and I think it doesn't need much comment. I mean, this is sort of the future, and the New Yorker saw it. And I was really gratified to see in the New England Journal last month, Francis Collins agreeing with this vision. So I'll just read to you. If everyone's DNA sequence is already in the medical record, it is simply a click of the mouse. Well, I got to tell you, it's more than simply a click of the mouse. To find out all the information you need, then there's going to be a lower barrier to beginning to incorporate that information into drug prescribing. So that's the vision that we actually have had for a long time at our place. How to get this information into the fabric of healthcare. I will say that it is possible to get genotyped right now at many common SNP sites. Many companies will do that. 23andMe is one of them. This is my 23andMe report. What you do is you spit into a cup, and 6 weeks later you get an email saying access your website, and they'll tell you what your genotype is at 500,000 SNP sites. And they'll give you a report which lists sort of 40 to 80 diseases. So for Parkinson's disease, this is the kind of report that you get. And I'm a little bit at increased risk for age-related macular degeneration. There's some green ones that are not shown on here that I'm at lower risk for. And you can actually download the entire file and look at it to your heart's content. So this is what the download looks like, this is what my genotypes are at two important loci for me. One is an atrial fibrillation locus, where actually I'm one of the 1.7% of the population that has two risk alleles for atrial fibrillation. A cynic would say, well, why don't you just take a family history? My mother has atrial fibrillation, but my father doesn't. And so I presumably got one of these for my mom and one of them for my father. So I'm hopeful that I won't get atrial fibrillation. And this is the 9p21, one of the 9p21 SNPs, and as you can see, I'm a heterozygote there. And, both my parents are in their late 80s and without coronary disease, so I'm not sure what to make of that. What I said before was that I'm not sure you want to incorporate any of that into your sort of routine healthcare, but there are pieces of that information that I would be interested in having my doctor use right now. And they're illustrated on this slide here. The FDA currently labels something like 2 dozen drugs with genetic information. They don't say do it, but they say, if you happen to know, or here's an explanation for some of the variability in response to drug X. Their drug metabolism pathways, the gene and the drug, and there are other genetic variants, and then there's a bunch of variants in tumors, which I'm not going to talk about. So this is the list from the FDA, and the ones in red are the ones that I have. So I want my doctor to know that when I go on Warfarin that I actually do have one of the V4C1 variants, and my Warfarin requirements are probably going to be lower than most people's. So that has immediacy that the disease prediction stuff doesn't yet have. And if you think that's bad, within a year or two, we're going to have the capability of producing tons and tons, doing stem to stern DNA sequencing, so for less than $1,000. What we're going to do with that information is an interesting debate. But one thought would be, well, it doesn't change in your lifetime, so have it done once, archive it somewhere, and when you need to know about something like CYP2C9 or CYP2D6 or whatever, then look. That's one model. I like this Jiffy Lube of genome decoding. I think that's sort of says it all. So you can read it in the Times or you can read it in Discover magazine. And right now it's possible to get sequence from the entire coding region of the genome. That's 1 to 3% of the genome for less than $10,000. It's called all the genes of the genome, all the proteins of the proteome. So all the exons are the exome. So exomic sequencing is something you're going to hear an awful lot about because there's tremendous potential there, and that's happening right now, largely generated by stimulus money. So this is the Vanderbilt Electronic Medical Record. This is a patient that I take care of, or I took care of until he died in his mid-90s of heart failure. So you can see, you see his diagnoses, you see his procedures, you see his medications. And my initial notion in the early 90s was that we would populate his, actually early 90s, early 2000s, sorry, we would populate his record with something like this that would come up when this is the face page of his record. So these are listed important genotypes, and they're all in blue because doctors have nothing else to do in clinic but to click on a blue link and read all about BRCA1 or CETP or ACE or whatever they want. And then I realized that doctors are too busy to do that. I certainly am. And so maybe the ones that are important would be listed in red. And I think I've come to the point where even in this brief talk, I've introduced you to enough alphabet soup that I defy anyone to regurgitate back the exact letters and numbers that I've used with some facility. And I live in this world. So I think the way it's going to have to work if we're going to have this kind of information to help drive drug prescribing is that the face sheet is going to look like this. It won't be there. And it'll be there when you write an electronic prescription for Warfarin. This person actually happens to be on Warfarin. The system will look at what the CYP2C9 and VESPA variants are and say this person should get 2.5 milligrams, not 6 milligrams, or whatever. So I think that that's probably the way it's going to have to work. And that's the vision towards which we're working, and the vision that Francis Collins enunciated. So what it's going to take to do that is listed here. I thought it was a good place to sort of just take a breath. I want to emphasize that when you do this kind of genetics in people, what you're left with is something like 9p21. And then you get to ask, well, what is the biology there? So to exploit genome science right now means not just finding stuff, but understanding the underlying biology and making people's health better. It will take information technology. I don't think there's any way you can talk about incorporating genetic medicine into the fabric of healthcare without information technology. So if the electronic record is really important to us, and it's sort of an enabling tool. And I think it's important to have patients who are engaged in this process for many, many reasons. There are huge ethical issues, and there are huge other kinds of issues. So the vision is that, in 2004, which is when I started to think about this BioView project, we were in a research mode. We're still very much in a research mode. And by 2020, I used to say 2050 on this slide, so I think 2020 is a better number. We'll be at the point of having markers that are part of the medical record. So we therefore created a clinical laboratory to ask the question how to get from here to here. The information that we generate in this laboratory, as I'll show you, has nothing to do with the patients for whom we deliver care, because there's a real disconnect between what we do clinically and what we're doing in this laboratory. But obviously, we would like to develop tools that will allow us to move to the bedside, and that's the point of the exercise. But right now, it's just a laboratory. The laboratory started planning started in 2004, and it took until 2007 before we started to collect samples. And I show you this slide not to review all the pieces, but to show you that it is a very complicated process to do this. You don't just sort of say, oh, let's set up a DNA data bank, let me write the IRB application, and we're done. That's a tiny, tiny part of it. And it is interesting to me that we started this way, with talking to the community and talking to many parts of the community, and then developing logistics, and then obviously developing the regulatory framework for this, and some of the scientific framework as well. So the model that we use is an opt-out model. OHRP, the people in Washington who tell us how to protect human subjects, have guidelines on the use of biological material that is about to be discarded in research. And the rules are that you can use those samples and the clinical information that comes with it, as long as the clinical information is de-identified. De-identified is a complicated word. It doesn't mean anonymous. It means that you would have to work pretty hard to figure out who that is. And some people are comfortable with that idea, and some people are not. So that's why we have an opt-out system. This is designated non-human subjects research. So you could say, well, we're just going to do it. And we're going to do it under the radar, and there's at least one institution in this country that is doing this kind of thing under the radar. They just collect samples with de-identified clinical information with IRB approval, but it's non-human subjects. We think we ought to be telling our patients, and I'll tell you about that in a second. So the first step was to de-identify the electronic medical record. I'll give you an example of how that works. It has been reviewed by many, 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 many committees. Arguably the most important of which was actually the Community Advisory Board. I can talk a lot about that. There's ongoing review of this, and there's some publicity to allow opt-out. But the most important publicity is this. This is a form that every outpatient signs once a year. I can attest to that because I get my health care at Vanderbilt. And the form includes above the signature line a prominently displayed statement that's blown up here that includes a tick box. So the only samples that we accept are samples from patients who have this form on file and who do not have a mark here. So the way it works is this is a manila file with a patient number on it. I haven't held a manila file in my clinic for at least 15 years because we do all this electronically now. And the first thing that happens is that it's put across, the record is put across what's called a one-way hash, and the clinical information is scrubbed of HIPAA, safe harbor identifiers, and other identifiers. So what a one-way hash is a mathematical function brought to you by your friends at the NSA, the cryptography community. It's a function that allows you to take this number and generate, in our case, 128 character hexadecimal code. And every time you give it this input, you get this output. Given this output, there's no way to go back to this. And this hash has never been broken, so there's never been anyone who can go from here to here. So that's what happens to the medical record number. The rest of the information is scrubbed in a way that I'll show you in a second. And that happened in the fall of 2006. And importantly, it can be updated. So nominally, we update every week. In reality, we have lots of things to do, and we don't update except every couple of months. So this is what a scrub record sort of looks like. It's not really the way it is, but it's a good example. Every date is changed. They're changed by the same amount within each patient's record. So if you get a medicine on Monday and have a rash on Thursday, that temporal relationship is maintained. On the other hand, the date shifting is by 1 to 364 days per record. So if it happens, if you're interested in studying a phenomenon that's linked to the calendar, seasonal asthma, heart failure admissions after Christmas, this is not the resource for you. The names are all changed, so the dates are changed. Every name is changed in the chart. And the way this works is you can buy commercial software to do this. I signed a purchase order for a $45,000 piece of software in my title as Assistant Vice-Chancellor for Personalized Medicine to do this. That was the second largest purchase order I ever signed. I'll show you the biggest one in a second. And this is the medical record number, and we supplement this with an extensive informatics effort of our own. So we look at scrubbing as over-scrubbing and as under-scrubbing. This is an example of something that is over-scrubbed. So here's filed through the Easter seals, and that is recognized as a name, so it's scrubbed out. So we recognize that, we go through the records, recognize that, and then put it back. So that's a separate effort that goes on in terms of improving the scrubbing procedure. This is the best one. The GI system is shows the exam is soft, non-distended, normal bowel sounds, and the software interprets this as it's the name of a place in the state of North Dakota. So it takes that. So we tell it to ignore that if it happens in the physical exam. So those are the kinds of things that you learn that I never thought I would learn along the way. But those are the kinds of things I've gotten pretty good at right now. So we now have this resource, and then a blood sample comes along. This is scanned, it's hashed, and then on this side of the hash we ask, do we have this sample already? Is this a person who is opted out? Is this a sample from somebody who's less than 18 years old? Because we don't do pediatrics. We didn't do pediatrics at the beginning. We're starting pediatrics in January. That's a process that Xen has taken about 1.5 years to put in place. And if it's eligible, then we collect the sample, and we actually extract the DNA. We had a big debate about whether we should extract the DNA or not. So the way it works is an investigator would have an idea. And there are lots of ideas that people have, and I can give you lists of those. The first thing that happens is that they sign a data use agreement saying, this is what we're going to do, this is the genotype we're going to do, we're not going to try to re-identify people. And then they get a list, and here are the cases and controls that I want. We're building, through our CTSA, in fact, portals that will allow people to interrogate the synthetic derivative and find cases and find controls. Part of it is going to be automated, and then part of it is sort of a Google search. Here's a list of 1,000 potential cases. Now you go through them and decide whether they're cases or not. You're familiar with the way Google search works, because you sort of look down that list of 20 things, and it takes about a nanosecond to figure out which entries you want and which entries you don't. And that's the idea. So it occurred to us that you could then do research that has nothing to do with genetics. This is chart research, chart reviews, year 2009. But if you want to go on and get the genetics, then you get fewer samples, obviously. There's more samples here than there are here. You do the genotyping that you said you would do, and you give it back, give the genetics back to us. That was the way we built the system. I thought it was pretty goofy to sort of incorporate this, but in a world where lots and lots and lots of people are having lots and lots and lots of genome-wide association and more dense genotyping, we envision a point really pretty soon where we'll be able to do some research without having to do additional genotyping. So that's a real plus for us. Right now, I don't have the number for this week, but this is where we were about a week and a half ago. 57% women. Other people who do this kind of thing find the same thing. Women have more encounters with the healthcare system than men do. Our population is 11% African American, so this is a relatively small number, but now we have, about 7,500 African American samples. That's a pretty big collection. Lots and lots and lots of ICD-9 codes. It turns out ICD-9 codes are not the way to find cases and controls. So let me tell you a little bit about what we've done with this resource, and then I'll stop. The first thing we did was a study to make sure that the sample handling procedures that we put in place actually worked. That the sample you got was actually married to the electronic record that it was supposed to be married to. We did that with under an IRB approval, so we could actually go back not to the scrubbed record, but to the real record. And I remember the day that I sat down with, my partner in crime here, I have many partners in crime. My biggest partner in crime is our chief of biomedical informatics. So we decided to do this experiment. We do the experiment, and we did one 384-well plate, do a gender check. That's genetic information that's in everybody's chart. And we had 8 mismatches. Okay. 8 out of 384 is too many, I said to the informatics guy. The informatics guy says back, well, you did the experiment to put confidence intervals around your estimates of whatever it is you want to do, so now you have confidence intervals. I said, I don't care, that's way too many. So we sat down and looked at all the records, and it turns out we grabbed blood samples on a day that bone marrow transplant clinic met. And we had 3 people who had gender mismatched bone marrow transplants. Remember, we get the DNA from circulating white cells. So that was interesting. And then a whole bunch of other people who had hypertransfusions, where the genotyping is okay, but it's not perfect, so the genotyping calls were not perfect. And then 1 real gender mismatch. And we said, well, it's 1 out of 384, we can live with that. So that's these kinds of experiments up here. The next thing we said was, well, if somebody wants to use this as a resource for their science, they would want to know that we can actually find stuff that we're supposed to find, like the 9p21 signal, for example, although that's not part of the experiment I'm going to show you. So what we did was we said, after we had about 10,000 subjects, we went to our CTSA, and asked them for money to support some genotyping, and we got some. And so we genotyped 150 high-value SNPs. High-value means that they are repetitively associated with disease and genome-wide association studies, or that they're somebody's favorite SNP. So CYP2C9 is on that list. CYP2C19, which is the gene where variants determine response to Plavix, another story that I haven't talked about, are on the list. A lot of neuroscience genes are on that list because we have a neuroscience group that's interested in this. And then these 10 diseases. And so I thought this was going to be the hard part, and the easy part is just finding the cases and the controls, no problem. Because when we started this, I really thought that Aetna or Blue Cross Blue Shield or Medco or somebody would have developed methods to excavate in electronic records to find cases and controls. And it turns out that is wrong. There is nobody who knows how to do this. The person who knows how to do this best right now in the year 2009 is a young faculty member at Vanderbilt who has spent the last 2 years developing the NLP methods along with others in a network that I'll describe to you in a second. So the first diagnosis we started with was rheumatoid arthritis. I'm an internist and I can diagnose rheumatoid arthritis, so I figured that should be no problem. And that turns out to be a real mistake, because what we learned is that you have to first of all talk to the guys who are in the rheumatology clinic every day, because they're the ones who can tell you exactly what it is in the chart, whether it's a piece of history, whether it's a set of medications, whether it's an ICD-9 code. In this case, for example, there's, when the biologics were introduced for rheumatoid arthritis, Enbrel and drugs like that, the rheumatology community was pretty convinced that they worked for psoriatic arthritis. The payers wouldn't pay unless the diagnosis was rheumatoid arthritis. So I leave it to your vivid imaginations to think about how many miscodings there are in terms of the ICD-9 codes. So the ICD-9 codes are useless for this particular diagnosis. They're okay for others. And so what we do is we develop the algorithms. Josh develops the algorithms, and then he deploys them in a large number of records. And then he'll get 100 or 200 records that his algorithm says are rheumatoid arthritis. And then a house officer or a fellow will look at them with a checklist and say this is or is not a case. So we review all those, and when we get a positive predictive value of 95%, then we say the algorithm is good enough, and we stop. And it took 4 iterations. The details of the iterations are shown here, but it took 4 iterations before we could settle on a set of patients who really had rheumatoid arthritis. And the same thing, you have to do the same thing for patients who really don't have rheumatoid arthritis. Somebody had to have gone to the trouble of asking. So a patient whose record consists of a broken ankle encounter in the emergency room doesn't count for any of that. So we ended up with 255 for sure cases, 1,184 for sure not cases, 507 possible cases that we would have had to manually curate. It turns out that what we did was we said, well, let's do the analysis with these, and then with these and these. And it turns out the results are actually pretty similar. And the natural language processing people are thinking about what that means in terms of the algorithms that they develop. So we studied initially 5 different diseases. They're shown here. Most of our cases are Caucasian, so the analysis that I'll show you is confined to Caucasians. We've done a very, very big ancestry informative marker study within this data set, so we understand the ancestry in our population pretty well. And that was a poster in Hawaii last week. So this is the data that I'm going to show you. 5 different diseases, 21 different SNPs. These are the genes that they're in or near. This is the SNPology. And these red dots are the odds ratios that one would predict for these variants from the literature. And these are where we are. So we're very, very, very gratified to say that every single dot for us is on the right side of this line. Some of these are not quite, they're all on the right side of this line. Some of these are tremendously underpowered. We would have to have about 3,000 cases. We only had about 500 cases of diabetes to begin to find this signal. But of the ones where the odds ratios are above 1.4, 1.5, we find most of them with statistical significance. And the others all trend in the right direction. So we're pretty happy with that. So we've thought about how to present the conclusion from that part of the work, and what we say is you set the study up with the null hypothesis that biobanks are useless, and so we now reject the null hypothesis, and we say that biobanks can be used to find patients with diseases. And so the next steps are opening the resource to the Vanderbilt community, which I'll talk about in a little bit, to do some studies not to find disease, but to find drug response phenotypes, and then something called PheWAS, which I'll tell you about in 1 second. So one of the things that we're most excited about is VESPA. my informatics guy is great at making up acronyms. So VESPA is his, BioView is his, and there's a couple of others that I'll tell you about in a second that are also his. It's pretty easy at Vanderbilt because everything has to have a V in it. So the VESPA project is basically the demonstration project that I just showed you for pharmacogenetics. Can we find drug response phenotypes? Can we relate them to things that we know ought to be there? And then can we start to look at unknowns? So we'll validate known, we'll discover new associations. And at the end of the day, we'll find a set of genotypes that would be a starting point for the Dan Roden, Francis Collins view of the world to put into the electronic record. And we think we actually know how we're going to go about doing that. But that's the desired outcome. This is a project that has been funded by stimulus money as part of the GO grant projects. This is a PheWAS. So a GWAS study is a genome-wide association. And the way those data are displayed, as I told you before, is chromosomal location on the x-axis and a p-value for each one of the comparisons you make on the y-axis. This is a phenome-wide association. So in a genome-wide association, you say, take people with a known phenotype, yes or no, and you look across genotypes. Here what we're doing is we're asking the question, people with a known genotype, yes or no, look across all phenotypes. Sounds sort of boneheaded, but I'll tell you why it's important. So this is not chromosomal location, this is ICD-9 code. So this is a SNP that has been associated with Crohn's disease, and Crohn's disease is one of the dots that pops up above the chatter down here. Prostatitis also pops up, so you ask yourself, maybe this SNP really is associated with prostatitis. This is a cool, the easiest way to explain this experiment is I have a friend who studies transcriptional mechanisms, and there's a common polymorphism in LXR, which is a transcription factor that's important for cholesterol metabolism. He studied it extensively in vitro in African American populations, and he writes me an email and says, what is the phenotype that would be associated with this variant? And I said, well, there's no way to answer that question except this, where you have a collection of patients with dense phenotypes, and you can ask this question. So we're sort of exploring that possibility as a hypothesis generating tool, just like genome-wide associations, hypothesis generating. And then I want to just say a little bit about numbers. And so this is, we've been interested in warfarin at our place. This is a study of warfarin that's in the literature, and basically the point of showing this is that in order to study, this particular study compared aspirin to warfarin in a clinic population, screening 4,600 patients with atrial fibrillation to study 900 and study bleeding rates. Bleeding rate is about 2%. If you look across populations, the bleeding rate for warfarin depends on age, and it can be as high as 10% in people over 80. If you want to know the genetics of bleeding, not the genetics of the INR, but the genetics of the bleeding complication, the largest study in the literature involved 50 cases of warfarin-related bleeding and looked at CYP2C9 variation. So if you want to study warfarin-related bleeding, you need a large collection of patients that includes the 80-year-old woman on 20 different drugs, and that is BioView. So we've been looking in BioView at cases of warfarin-related bleeding. So this is a portal that anybody with a Vanderbilt ID can go to and just enter some ICD-9 codes and other search criteria and get a number. You can't look at records, but you can get an idea of whether we have a lot of cases or not. In this particular analysis, we had 4,842 patients who were taking warfarin and who had DNA. So that was about a year ago, actually 6 months ago. So we started with that number, and with natural language processing, identified 1,275 possible warfarin-related bleeds, 932 of whom were admitted to hospital, and we curated a subset of those and found that 124 were admitted to hospital for warfarin-related bleeding. And so by extrapolation, there are around 372 real cases. That was in April of 2009, so by now we should have over 500 cases. That's 10 times as many as the largest studies that looked at warfarin-related bleeding, and that's at one center. So that just illustrates the potential of a resource like BioView. So let me just tell you about VEGR, another acronym that my bioinformatist has made up. VEGR stands for the Vanderbilt Genome Electronic Records Project, but if you're a Trekkie, you will know that VEGR stands for the Voyager spacecraft that they were pursuing in Star Trek I. So VEGR is one of 5 projects that part of the eMERGE network, the Electronic Medical Records and Genomics Network. We're one of 5 sites. We have more samples at our site than all the other sites put together. This is supported by the NHGRI, and the goal is to assess the utility of the kinds of resource that we've built. The carrot for us is that we identify a phenotype of interest, and they will support a genome-wide association analysis, the genotyping for a genome-wide association analysis. I can't resist saying that I got an email this morning that our data, our genotyping data was actually delivered this morning. So I hope that our analysts are hard at work trying to figure out if we have a signal for the phenotype that we chose to study. But the outcome at the end of the day will be around 20,000 subjects with dense electronic medical records and genotyping information. So if you want to know the genetic component to serum calcium, you want to know the genetic component to platelet count, you want to know the genetic component to GFR, to some electrocardiographic measure, to hypertension in blacks, to cataracts, to HDL, all of that is there. It's hard to get at, but it's there. So widespread use by VU investigators. So one of the biggest limiters of this particular project in making these samples available to the community has been robotics. You have to, if the demand is 1/10 of what we think it might be, then you have to have robotics in place to access the samples, to aliquot the samples, to keep track of the samples, and all that. So we applied to the high-end instrumentation grant program at NIH. We were successful in that, and so we finally figured out where to put this robot. So about 2 weeks ago, I signed the largest purchase order that I've ever signed. I got a special pen to do that, as you can see. And that was $1 million for a robotic system. It's $1,982,000 for the RTS robotic system. So we, in collaboration with our CTSA, have built a portal that will allow investigators to not only to start to look at the synthetic derivative and identify cases and controls. They can do that right now. Because it's non-human subjects, they still have to apply to the IRB, but the IRB says it's non-human subjects, it's expedited, so they get a review in 2 or 3 days. The samples will be released probably, we could do it like the middle of December. We think that that's probably a bad time of year to do that. We'll probably do it in early January. The first releases won't be released at all. We'll do the genotyping ourselves just to make sure we have the mechanics of it done right. But we're very excited about the idea that we're going to make this available to lots and lots of people. The kinds of questions that people ask are amazing, but sort of from things like, do you have a sample of 1,000 African Americans? We don't care what the phenotype is, we just want to know what their ethnicity is, to complicated drug responses like hypotension during delivery. So that's a complicated phenotype, but we could probably do all of those things. So big challenges. This is a real-world resource. So there are 80-year-old women with 20 drugs in the resource. I like to use the word parallelizing, decreasing the time and cost to generate sample sets. It's not zero, but it goes pretty quickly, especially now that we're learning how to do it. And I think that if you're buy into the idea that the electronic record is something you're going to have to do to incorporate not just genomic science, but high-dimensional science, all kinds of omic science into practice, then we're learning how to use that electronic record. And the complexity of the data sets are both a challenge as well as an advantage of a set like this. And I just listed some of those there. Here's another part of personalizing medicine that I thought it was worth saying something about. I communicate with my doctor through this portal called My Health at Vanderbilt. This is me again. And when my page comes up, it comes up with information for me. So this is no secret, I have hypertension. So it comes up with a little hypertension thing. My wife, it comes up with different things because she has different kinds of healthcare things. And the cool part of this is that this thing over in the corner. It's not a great way of doing it, but we're interested in the idea of talking to patients and finding out if they have when they start a new medicine, particularly medicines that are newly marketed, do they have new experiences? This is not a great way of doing it, but it illustrates the idea that you can actually engage patient populations. And we're not the only ones to think about how to do this. There are others around the country who think about how to do that as well. So final thoughts. The data sets are important, but at the end of the day, we want to use this to figure out how to bring it to the bedside. Informatics is hugely important. Statistics is hugely important. And all this GWAS stuff that you've been hearing about for the last 4 years is really interesting from the epidemiologist point of view, but at some point the basic scientists are going to have to bite off what all these biological signals are. And that's the real challenge and the real fun part of this. There are other dimensions to this that I haven't talked about, but have to be part of a comprehensive program: ethics, economics, and international and national collaborations. We don't want to be the only people doing this, because I think that if you could imagine that there were 10 resources like this around the country, you'd have a national biobank immediately. So we have a lot of international collaborations and national collaborations. I show some of them on here. Some of them I've talked a little bit about. This is eMERGE. This is the Pharmacogenetics Research Network. These are other networks that I'm fortunate to be part of. I believe in that kind of science. And these are the people who do all the work. This is Dan Masis, my partner in crime, the chief of informatics at Vanderbilt. These are clinicians, managers, biostatisticians, genetic epidemiologists. I'm trying to think of who else. Ethicists. Information science, privacy wonks, natural language processing people. And I want to say a word about these guys down here, now that the dean has left. When I dreamed up, so the person in the middle is Roscoe Robinson, Ike Robinson, our former vice-chancellor, who passed away a couple of years ago. He was vice-chancellor since the mid-'80s. And in 1991, he said, I want Vanderbilt to be the nexus of information science in healthcare. So that's when we started our electronic record, and it was due to his vision that we have the resource that we have now. This is Harry Jacobson, who succeeded him in that job, running the hospital and the medical school. And I went to Harry in 2004 and said, I think we should create a DNA data bank. And a reasonable answer from a person like that is, well, generate a 50-page document and a 10-page Excel spreadsheet with all the projections for financial implications for this kind of project, and then we'll talk. And Harry said, no, I think we should do that, and we had a handshake, and that was the beginning of BioView, so a visionary. And then this is our new Vice-Chancellor for Health Affairs. He also buys into personalized medicine. He actually worked in Harry's office to get BioView running. He's a very talented ion channel biologist and my first graduate student, so I hope it'll work. Jeff Balzer has been in the job for about 6 months. So institutional leadership is really important in all this. Thank you, and I'll be happy to take questions.